Hello everyone, welcome back to my YouTube channel Biology Exam Preparation with Nilesh Anandwani, a YouTube channel for all the students of biological sciences. Either they are preparing for the CET examination, NEET examination, IIT-JAM examination, GATE examination, PET examination, SET examination or UGC CSIR NET examination. Welcome back to my YouTube channel. I am a Professor Nilesh Anandwani. Today I am here to discuss with you the revision of Chapter 11 from the 12th CBSE, that is Biotechnology, Principle and Process. In this chapter, I have delivered around 8 to 10 lectures, and in this 8 to 10 lectures, I have completed the complete course of this Biotechnology, Principle and Process. Complete chapter we have discussed. But in this lecture, in just a small video, I would like to give you complete revision about the chapter from upper point, from highlighting words, highlighting concepts. In this chapter, we started with a basic point, that was introduction to biotechnology. That what is biotechnology? Biotechnology is a branch of a biology or branch of a science which deals with the study of applications of technology in the field of biology. That was called biotechnology. Applications of technology in the field of biology, that was called biotechnology. We discussed so many products of biotechnology, like in the 12th century, Portugal people used to produce wine from the grapes. Nowadays also wine is almost produced from grapes and as well as from other products also. Portugal people used to collect the fresh grapes from the farm, and they used to jump on these grapes and they used to extract the juice from it. That juice was allowed to ferment with the help of a Saccharomyces cerevisiae yeast we called, or commonly we called baker's yeast, which undergo fermentation process and finally produce the product. That product is a wine. Alcohol, wine, beer, champagne, all these alcoholic beverages are product of biotechnology. In introduction point, I taught you several examples of biotechnology, like a genetic engineering, means rDNA technology, tissue culturing, cloning, DNA fingerprinting, southern blotting, northern blotting, western blotting. Several techniques I taught you about the biotechnology. Even though protoplasm fusion and cytoplasmic fusion, test tube baby in vitro fertilization. Test tube baby in vitro fertilization is same thing. Cloning, human cloning, so many examples I taught you about the biotechnology in introduction. Second topic we discussed, you can say principles of biotechnology, or you can say principles of genetic engineering, or principle of genetic engineering, or rDNA technology. rDNA technology is basically based on collection of a particular donor, removal of particular gene from that donor's DNA, keep the genes safe, transfer these genes into the vector. Vector means plasmid. Transfer this recombinant, plasmid plus interested gene is called recombinant, into the you can say competent host, and after transfer that competent host into, you can say, small scale production, you can say culturing of that bacteria, and after culturing just grow that bacteria at the large scale production. And once you get the large scale production, you will get large amount of product, might be your insulin or whatever. To understand this rDNA technology in detail, we have gone through the several topics. Like a first topic we discussed, that was restriction endonuclease. So what was restriction endonuclease? Restriction endonuclease was the enzyme which was responsible for cutting a DNA at a specific point. It was first time discovered by the scientist called Werner and Erber. Okay, Werner Erber basically, Werner Erber was a scientist, and this was helped by the other two scientists also. We discussed restriction endonuclease in several. Restriction endonuclease was belonging to large class called as a nuclease, which was of two category: endonuclease, exonuclease. Exonuclease basically cut the DNA top or at the bottom. Okay, and endonuclease cut the DNA within the DNA, inside the DNA. That was endonuclease. Then endonuclease was of three category: type 1, type 2, type 3. Type 1 and type Third, are used generally in our biological process, whereas type second are you can say used in the genetic engineering in vitro experiment. We discuss basically nomenclature of enzyme, means how to give a name. First restriction endonuclease enzyme was hind second, means H means hemophilus, IND stand for influenza, hemophilus influenzae restriction endonuclease second. Then we discuss some example, Eco R1 Escherichia coli restriction endonuclease 1 or strain 1. I told you restriction endonuclease BAM second, like a Bacillus amyloticus second. Then we discuss cleavage, you can say recognition sequence, means how to find out which segment has to be cut in the DNA. That we studied that it should be the 6 nitrogen bases, it should be mirror image to each other. It will be called as a palindrome sequence, means if you read from right to left or if you read from left to right, it will be mirror image to each other, like a Malayalam. If you read this word from right to left or if you read this word from left to right, you will get same. That will be Malayalam to Malayalam only. Okay, this is called mirror image or also known as a palindrome sequence. Okay, then we discuss cleavage pattern, that how to basically cut the DNA. When we are interested to cut the DNA, only restriction endonuclease we don't have to use. We have to use you can say DNA phosphatase also, just to mark, and marking is done by the magnesium ions. Okay, I also taught you some enzymes which are used in the genetic engineering, like a restriction endonuclease I taught you. Helicase enzyme used to cut the DNA but at a free nucleotide, top and bottom, but not at a specific location, it can cut at a random position. Then I taught you DNA phosphatase, which is responsible for marking the DNA where to cut. Then I taught you DNA ligase, which is joining the DNA molecule, RNA molecule, nucleotide, nucleoside, genes, chromosome, etc. Two genetic molecule can easily be joined by DNA ligase. Then I taught you about DNA polymerase. It is used to convert nucleoside into nucleotide. It is responsible for converting two single stranded DNA into double helical structure. So like this. And basically reverse transcriptase found in HIV virus used to convert single stranded RNA into double stranded DNA. So by the process called reverse transcription, and so the name of enzyme is given as reverse transcriptase. Then I taught you about polymerase chain reaction. It was invented by the Carey Mullis, and his full name is Carey Banks Mullis. This Carey Mullis introduces this technique. Basically he introduces this technique just to increase the size of DNA, multiply the number of genes. It is used in the research field, medical field, number of area, forensic science, crime branch, etc. Number of applications was PCR was there. PCR was basically requiring few things, like a deoxynucleotide triphosphate, that's called DNTP, thermal cycler means PCR machine, then primer used to attract nitrogen base, then DNA sample to whom we are interested to increase size, and last but not the least, yes, what is that? Are you forgetting? Take polymerase, thermus aquaticus polymerase enzyme, which is main hero of our PCR. In PCR there are 3 steps. Like in first step we are heating the DNA sample at a 95 degrees Celsius, that's called heating or heat denaturation or melting, where nitrogen bases get separate due to the breakdown of hydrogen bond. Then in second step we are joining the DNA molecule by the deoxynucleotide triphosphate DNTP as well as DNA primer, where the step is called cooling or annealing. We are reducing temperature from 95 to the 55 degrees Celsius, that's why we call cooling. Whereas in last step we are increasing again temperature from 55 to 72, that's called polymerization, extension or elongation, where Taq polymerase is converting single stranded linear DNA molecule into double stranded DNA. Carey Mullis was awarded by Nobel Prize for this technique in the year 1993. Then we discuss about plasmid vector. What is plasmid? It is a circular double stranded automatic replicable extra chromosomal DNA. Plasmid is also circular, but plasmid is a small circular double stranded, and it is extra chromosomal DNA. It is present along with other organism. First plasmid was introduced by the Joshua Leder work, and there are so many plasmid basically discovered up to now, like a PBr322 plasmid discovered by Bolivar and Rodriguez. PBr322. PUC plasmid discovered at University of California. R plasmid, drug resistant plasmid. Then Cole plasmid found in E. coli bacteria, and then kappa particle found inside the yeast. TI plasmid found in Agrobacterium tumefaciens. All this plasmid we have studied in detail. We understood that how plasmid act as a vector for transferring genes, because plasmids are self replicable, means automatic replicable. This is the best feature of them, which make them powerful to consider as a or ideal vector amongst the all other vector. There are some artificial vectors are also available, such as a bacterial artificial chromosome, YAC, yeast artificial chromosome, HAC, human artificial chromosome. Then we discuss about electrophoresis. What was electrophoresis? Electrophoresis is a technique by which we separate the DNA molecule, RNA molecule, protein molecule, or any biomolecule on the basis of its charge. If a molecule have a positive charge, cationic charge, so it will move towards anode. Because of positive charge, molecule will move towards negative charge, and if on self they possess negative charge, anionic charge, so they will move towards cathode, positive charge. So likewise there is a positive and negative charge is there. In electrophoresis we discussed the gel which we are using is agarose gel. What we have to do, first put the agarose powder into the flask, or you can say beaker, and then put a water and just heat it. After getting liquefied that powder, just transfer that liquid into the electrophoretic machine or electrophoretic apparatus, and let it dry for some time. After letting dry, just load the DNA, RNA or protein sample into the vial, actually, and then supply the electricity. After 1 hour, you will find some strands of DNA or bands of DNA, but you will not be eligible to see that DNA by your naked eyes. You have to stain that gel by the ethidium bromide, and once you stain it by ethidium bromide, you can see that, but you have to use a UV light. When you will see this gel into the UV light, you will be able to see the DNA bands, and when you wanted to separate this, remove this DNA bands from the gel, you have to process, you have to do the process called elution. Elution is a process by which we remove the DNA from gel. Then after electrophoresis, we move towards a gene transfer, that how to transfer gene. Gene transfer can be done by very easily. It's not that much easily, but understanding concept is very easily. When you wanted to transfer directly genes into the plant wood, as we can see, this is a plant. When we want to directly transfer genes into the plant, so we have to carry the genes, and cover it either by tungsten or either by the gold particle, and then shoot it with the help of a gene gun or biolistic. It will directly enter inside the stem. Genes will remain inside the stem, and gold particle or tungsten will remove it out. So genes will remain inside it. That is called gene gun or biolistic. Then we can use a micro injection, directly put inside the nucleus. When we want to transfer genes from one bacterial cell to another bacterial cell, we can use a technique called that's called as a transformation, or Griffith experiment we studied. When we want to transfer genes from one eukaryotic cell to another eukaryotic cell, we can use transfection. And when we want to transfer gene from bacteriophage, bacterial virus to bacteria, we can apply transduction. So these are some methods of gene transfer mechanism. Then we discuss about step-by-step method of rDNA technology, means procedure of rDNA technology. I told you rDNA technology is basically done by the 6 steps. Like first we have to select the donor, then we have to remove the DNA from the donor, isolation of donor and removal of DNA, that is first step. In second step, we are cutting the DNA with the help of a restriction endonuclease enzyme. In third step, we are cutting the vector, means plasmid, with the help of same restriction endonuclease enzyme. After that, we are in fourth step, we are transferring in third step only we are transferring interested gene into vector where we have cutted, and this becomes R, means recombinant. We are transferring this recombinant into host cell in the fourth step. Insertion of recombinant into host cell is the fourth step. And finally, in fifth step, we are culturing the bacteria and extracting the foreign gene product. And in the sixth step, we are increasing the quantity of our desired product with the help of a large scale fermentation process. This is sixth step. First step, isolation of donor DNA. Second step, cutting of DNA at specific location by restriction endonuclease enzymes. Third step, cutting of vector at specific location by same restriction endonuclease, and then mixing this interested gene into the vector in third step only. Fourth, transferring this recombinant, means interested gene plus vector, transferring this recombinant into the host cell. Fifth step, culturing this host cell and allowing the bacterial host cell to increase in number but in small scale. And in last step, sixth step, we are growing this bacteria in a large scale fermenter bioreactor, so that we will get large amount of a product, and that product is essential and transferred to all over the peoples, those who need that product. Like a human insulin, first time it was produced by rDNA technology as a humulin or human insulin by the Genentech company from the American based, and selling was allowed to the company called Eli Lilly and company from the you can say United States of America. Then we discuss finally upstream and downstream process. What was that? Upstream fermentation process was beginning from the collection of raw material up to the production, and downstream process was related from collection of final product up to the selling of product. That was downstream process. In upstream process, we used to collect the raw material and put into the bioreactor, then prepare a inoculum, means organism, and put into you can say fermenter, then put a media, nutrient media into fermenter, and go for the you can say process we call bioreactor process or fermentation process. This is up to the upstream process. And finally, in downstream process, after the completion of fermentation process, we are collecting the product, we are purifying the product, we are testing that product with the help of a quality control QC, quality analysis QA, and research and development R&D. And finally, when we are getting okay sign that our product is a good to the human consumption, there is no problem to the human after consuming this product. After getting this sign, we can go for the packaging of product, labeling of product, and after labeling product, we are ready to sell this product, launch this product into the market. This is downstream process. So upstream process, downstream process, both are very important in the fermentation process. So this was our complete chapter called Biotechnology Principles and Application. Dear students, I would like to tell you few highlighting points. In this chapter, there are so many you can say chances of asking around 3 MCQ, and these MCQs are from one MCQ is from the PCR or gene transfer. Yes, one MCQ is guaranteed from the plasmid, because structure of plasmid explained in CBSE is a large, and one MCQ is definitely from the process of rDNA technology. So this 3 point has to be considered for the exam point of view, is very important. No doubt our complete chapter was valuable, and I have took this chapter in a frequent manner that every topic you can easily understand. If you have any kind of a doubt, what I have taught you in this revision was from highlighting point, upper point, but if you wanted to understand all this point in detail, you can basically see my YouTube channel. In YouTube channel, this chapter, all the videos, around 10 to 11 videos are there. related to this chapter. And if you wanted to see a complete chapter in one video also, so just wait for a few days, like 2 to 3 days only, and within 2 to 3 days a new video will be launched which will cover introduction up to the MCQ paper. Yes, and after this video, next video, we are going to discuss about MCQ practice of this chapter. So thank you so much for watching this video. If you found that this lecture was valuable to you, you got some knowledge, then please hit the like button. And if you want to see such kind of more and more video, then please subscribe to our channel and press the bell icon so that you will be notified when I will come online or when I will upload my new video. Thank you so much. Have a good day.